Okay, so we are going to continue with the process of fertilization, and today we are going to learn about the events which take place during this process. So last time, in our last lecture, what we learned was about formation of gametes, how oocyte and spermatocytes, they get specialized. Today we are going to see the process of fusion, which is referred to as fertilization, which is shown in these drawings I took from Scott Gilbert's book, Developmental Biology. So this is just the point where you can see a little hump, the entry of sperm nucleus into the oocyte. And then after this event, you can see cell, so the nucleus of sperm that is released inside the oocyte, which fuses, which means, after this event, this completion of second meiotic event takes place, and cell enters into meiosis. You can see here the spindle fibers, and eventually one cell will start dividing into two. Now, in order to understand this process, we have to understand the structure of sperm as well as the oocyte. And also we have to learn about what are the contents in these specialized cell types. As I said, and we learned in last time as well, that both sperm and oocyte, egg cell, they are very specialized structures. Sperm, for example, all sperms are motile, and the entry of sperm into the oocyte, that's what leads to activation of the egg. And this activation of egg means the meiotic arrest is, unblocked, and cell completes the meiosis. The sperm cell has a nucleus, which is shown here. You have acrosomal vesicle. We are soon going to see what is the function of this acrosomal vesicle. This is actually something very specialized thing which provides selectivity or specificity for species that, a human sperm will only fertilize a human oocyte, and mouse sperm will oocyte only a mouse oocyte. Then you have mitochondria, which provides all the energy for the motility of the sperm, and it helps in the penetration of sperm into oocyte. And then you have this long flagellum, which is this whip-like structure, which helps in the motility of the sperm within the reproductive tract of females. In case of oocyte, here this actually drawing from Walpert is describing all the steps in the process of fertilization, 3, then step 4. Oh, sorry, where's my slide? Do you see my slides?
Yes, sir.
Then you have, sperm nucleus entering into the oocyte. So we will see what structures or what are different features in the oocyte which provide obstacle or barriers, as well as facilitate this process of fertilization. So the movement of the sperm into female reproductive tract and then eventually fertilization, that process is called capacitation, process of capacitation. Now, if this is the oocyte, you can see this a layer of cells, and this layer of cells is called cumulus cell layer. This is first barrier to the entry of the sperm. And the cumulus cell layer is present in a sticky mass of hyaluronic acid. And the enzyme is hyaluronidase is present in the sperm head, which facilitates this disruption of the cumulus cell layer. And by the action of this hyaluronidase, after disrupting the cumulus cell layer, eventually the sperm start entering into the oocyte. Now, this is the first obstacle. Then the second obstacle is the zona pellucida. Zona pellucida is this region. You can see this is actually made up of 3 glycoproteins. So this layer of glycoproteins is also another physical barrier to the sperm entering into the nucleus. Now, this is the place which provides species specificity, where sperm cell gets selected and it is ensured that this sperm actually belongs to the right species and it must be allowed to enter. Now, there is ZP3, zona pellucida 3 receptor, and then with the help of that receptor, this reaction takes place when sperm arrives at the zona pellucida. So inside the sperm, so the contents of the acrosomal vesicle, I told you it's a very specialized structure which provide the species specificity. So these contents of acrosomal vesicle in sperm contain SED1 in the sperm head that binds to the zona pellucida ZP3 receptor, and acrosin protease, which is shown here, is being released, and sperm then passes through the or penetrates through the zona pellucida. Then comes this membrane, and when sperm passes through the zona pellucida, eventually the membrane of sperm fuses with the plasma membrane of oocyte, and nucleus is released into the cytoplasm of oocyte. You have here the oocyte nucleus. Now, when it enters, there is still another, I call this precision design or engineering. You see these blue granules, which are written as cortical granules. Now, they are very important. As soon as you can see here, when the fusion of sperm cell membrane and oocyte plasma membrane fuses, these cortical granules, they come outside the cell membrane. You see, they are now no more inside. They, like exocytosis, they come out of the plasma membrane, while the sperm nucleus is released inside the oocyte. Now, what is the role for release of cortical granules here? The purpose of cortical granules is because only one sperm can fertilize. There are hundreds and thousands of sperms are traveling through the reproductive tract of females, but there's one oocyte and only one sperm is eventually going to be successful. As soon as this acrosomal reaction takes place and then eventually this plasma membranes get fused, the cortical granules are released. They become outside the plasma membrane, which now tell all the incoming sperms that the oocyte is successfully fertilized. So after the acrosomal reaction, you saw that the sperm surface proteins, they get exposed to the which is basically, this is the acrosomal reaction. All the sperm proteins and whatever is present in the acrosomal vesicle, they get exposed. They are released to the zona pellucida, which facilitate its movement through the zona pellucida, and eventually nucleus is released. There is a protein called fertilin that binds the integrin like egg receptor. Sorry. All is happening at this acrosomal reaction. Okay. Now, there is, how do we learn that the process of fertilization has taken place? And, you know, the cortical granules are released, etc. The cortical reaction at fertilization, this experiment was done in sea urchin. And as I told you, the release of cortical granules, they eventually form a membrane outside the plasma membrane of the oocyte. Now there is, as soon as the sperm cell membrane fuses with the oocyte cell membrane, there is membrane depolarization. And when membrane depolarizes, there is change in the potential, and that can be measured. That was done in the sea urchin. Here you can see the potential measured in millivolts. So there's no change in an unfertilized egg. So it's, as soon as the sperm is added and the process of fertilization initiates, you see, due to membrane depolarization, the potential of membrane changes, and it comes nearly minus 15 to plus 15 to plus 15 to plus 20. And then again, when membrane, you know, so the sperm nucleus is released, the membrane depolarization is gone, and you see the membrane potential is going down. Now, as soon as you add sperm, this is the time when cortical granules are released, which is an indication of fertilization. And then eventually these cortical granules, they form a fertilization membrane. We call this fertilization membrane because the cortical granules, which are released, which are now outside the plasma membrane, they make a layer which is called fertilization membrane. Now, yeah, so this was about the electrical potential after the membrane depolarization. We will see another indication of process of fertilization. So this is what is happening, which is being measured in case of electrical potential. So you have here a vitelline membrane, then this is the plasma membrane. The outside this dotted line is the vitelline membrane. And within the cell, you have these cortical granules. Now, as soon as the process of fertilization takes place, there is a wave of calcium ions. And soon we are going to see uh, what are the consequences of calcium ions, and are these calcium ions, or this wave of calcium ions, is important. We are going to see in next slide. But you can see, so after the calcium ion release, which is an evidence of fertilization, these cortical granules are exocytosed through the process of exocytosis. They, instead of residing within the cell, uh, they are moved outside the cell, and between the vitelline membrane and the plasma membrane, you can see these cortical granules. They are arranged in such a way that you have a membrane-like structure, which is called fertilization membrane. Now, this fertilization membrane is important because they block the polyspermy. They are an indication that one sperm has fertilized, and they prevent any other sperm which may be trying to fertilize. So that's a indication to rest of the sperms. They then die away. Okay. Now, we talked about calcium wave, and this is imaging of calcium ions. What do you see here? This is 15 seconds after sperm nucleus enters into the oocyte. And you can see here, these red ones are actually the calcium ions. Now, within next 4 seconds, and I would say in total 31 seconds from this point to this. So you can see the wave of calcium ions spreading throughout the oocyte. It's just like a big waterfall, a big explosion taking within the cells. And this actually is not only indication of the process of fertilization, but also it plays an extremely important role in eventually bringing this fertilized egg into the cell cycle. Now, you can think of experiments which proves that calcium ions are really important for process of fertilization. So we call this activation of egg. So this calcium wave is actually an indicator of egg activation, that fertilization is taking place. You can prove that calcium ions are very important by simply injecting EGTA. EGTA and EDTA, they are chelating agents. They chelate divalent cations, calcium or magnesium, etc. So when you will block, you will add EGTA into the oocyte and then allow sperm to enter. EGTA is going to chelate all the, so chelation of calcium ions, that blocks the process of fertilization. You don't see the cell entering into cell division mode. And as I told you, this wave of calcium or calcium ions, they're very important for proteins involved in cell cycle to bring this newly fertilized egg into mitosis. You can also activate egg by simply injecting calcium. Okay? And then you can mimic process of fertilization as well. Let's see what is the role of calcium here and the immediate events which lead to cleavage divisions, early rapid cell divisions. So there are, cell cycle depends upon cyclins and cyclin dependent kinases. Um, so there is a factor which we call maturation promoting factor. So this maturation promoting factor, MPF, it must go down to complete meiosis. So we all know, we learned in our last lecture that the oocyte is arrested in meiosis 2. Okay. Now, this arrest is due to MPF, which keeps this cell arrested. This has to go down. This has to, its activity should go down so that this arrest is released and cell enters into cell cycle. So if you look here, this is the immature oocyte. Okay. Then you have this progesterone stimulus, which takes place at puberty. Then there is completion of meiosis 1. You see here. After meiosis 1 takes place within the, I was wrong, I said puberty. Meiosis 1 takes place when the females are within the mother's womb. Now, this completion of meiosis 1 takes place, and then oocyte is blocked or arrested in metaphase of meiosis 2. So they enter into meiosis 2, but they are arrested in meiosis 2. And this is the stage when we call this mature oocyte. Now, when the sperm enters the oocyte, when the sperm fertilizes, this arrest of meiosis 2 is released. You can see now completion of meiosis, and you have male and sperm pronuclei within the same cytoplasm. Now, when the fertilization is taking place, this arrest of oocyte is released. Meiosis is completion of meiosis takes place, and that is what coincides with the wave of calcium. So this wave of calcium leads to degradation of MPF activity. So loss of MPF activity, it brings down MPF, arrest is released, process of fertilization is complete. Now, again, it goes up, MPF goes up again. Okay. But every time when cell has to divide, you can see it comes down. And cell keeps dividing mitotically this way. And this one is indeed the 1n plus 1n. This is diploid zygote. Okay. So how calcium acts as it is written? So increased concentration of calcium ions that leads to activation of colmogylin dependent protein kinase 2. It's also called CAM kinase 2, colmogylin dependent protein kinase 2. And that is what leads to degradation of MPF here. So every time you are going to enter into meiosis, you have to bring down MPF, and cell division takes place. So we come back to fruit flies and try to see how the oocyte formation takes place in the fruit flies. I wanted to have this slide in the last lecture, but I missed it. So what you have is you have a diploid germline stem cell in the germarium, which is here. So this is the germarium of fruit fly. This diploid stem cell, it divides asymmetrically, which means one of the cell remains stem cell and the other cell, it becomes what we call cystoblast. Cystoblast. This cystoblast, it undergoes 4 cell divisions, okay? And eventually it produces 16 cells as a result of 4 cell division. And these cells are connected through cytoplasmic bridges, and these groups of cells are called germline cyst, okay, which is here. So a stem cell divides asymmetrically. One remains stem cell, the other one becomes cystoblast. This cystoblast, it divides 4 times to produce eventually 16 cells. And these cells are connected through cytoplasmic bridges. And collectively here, what you see outside this layer of cells, the outer one, they are called the follicle cells. They are diploids, and these ones are the haploids within. Now, out of these cells, one of the cell becomes the oocyte. All the other, they enter into polyploidization. They are polyploids. They are no more diploid. They enter in a very specialized cell cycle where they become, 4N, 8N, etc. Now, the purpose of these cells, what we have learned is that they are factories of large-scale mRNA productions. And these cells are called nurse cells. Nurse cells are the ones which take care of which nurse the oocyte, and they produce these large amounts of mRNAs which are deposited in the oocyte. And you should know which are these mRNAs. What are these mRNAs which are being deposited here? Are you all there?
जो maternal genes होती हैं bicoid.
Yes, these are eventually the maternal genes. Okay, you see here these much bigger cells, which are these nurse cells, and eventually the nucleus of the oocyte, which is here in the posterior side, it moves towards the anterior of the oocyte. This is the egg cell, which eventually is laid by the females after mating. Now, if you pay attention, this was the germarium, then this is the gradual journey. So when you have here egg cell being matured, you have a continuous production like this. You will have oocyte at this stage, you will have another oocyte at this stage, and oocyte at this stage, and they keep moving like this. Eventually, this egg is laid, and all the development in fruit fly takes place outside the mother's womb. These follicle cells, they play a very important role. Look, these are the posterior follicle cells, and these are the anterior follicle cells. And here, there's no distinction between anterior and posterior follicle cells. They are very important because these cells, they do talk to this cell and they talk to this cell. So there is a communication going on between oocytes. So yeah, I told you already that diploid cells, the somatic cells, they make the sheath of follicle cells around the nurse and the oocyte to form a chamber. And follicle cells are further subdivided into functionally different populations at various locations. This is what I told you. Anterior follicle cells and posterior follicle cells. We are soon going to see what they are doing in a developing oocyte. So this is this stage you just saw, and this is the oocyte location, nucleus of oocyte location, so location of oocyte nucleus. And here you can see large nerve cells. I think they are visible to you, all of you. Okay. Now, this is what the communication between posterior and the anterior follicle cells is taking place. And this communication is very important, not only for proper maturation of the oocyte, but also movement of nucleus, which is here near posterior and to eventually come here in the anterior pole. So first sign of the oocyte formation is actually that the nucleus, it from center, it moves to posterior and here in a developing egg chamber. Then there is rearrangement that takes place while egg separates from germarium, and due to the preferential adhesion between future posterior end of the oocyte, uh, future posterior of the end of the oocyte is this one or this one, and the adjacent follicle cells, which are these ones. Okay, you can see these different colors. We are soon going to see what kind of communication is taking place. So there is cell signaling going on. So there is a signal from the anterior of older chamber, which is this one. This is the older chamber. Okay. So these follicle cells, which are present at the anterior of this egg chamber, they send a signal to the posterior of younger chamber. And two signaling pathways are very important. One is delta notch. Notch delta is delta is the ligand and notch is the receptor. They specify the follicle cells to become specialized follicle cells. So these cells, they become specialized follicle cells. Similarly, this egg chamber also received signaling when this was at this stage, okay, or this one. So this communication is continuously taking place. Now, these follicle cells and the specialized anterior follicle cells, they send a signal to adjacent follicle cells via JAK-STAT pathway. JAK-STAT pathway, this is involved in stalk formation, which is this place, the green one. This is stalk formation is taking place between two independent egg chambers. So signals then from the stalk, young egg chamber, oocyte and posterior follicle cells, they are all they're all responsible for expression of EEC adherin at this stage. And eventually what we see in the older egg chamber, we see movement of uh, egg nucleus from posterior to anterior. So you can see this is shown here, how it is taking place. There's a protein called gherkin, uh, which is responsible for this movement of nucleus from posterior to anterior. It's a TGF alpha cytokine, and gherkin is expressed right here at the uh, end of the oocyte in the plasma membrane. You have then the follicle cells, which are having the receptor for gherkin, which is called torpedo, which is a EGFR, EGF receptor. And this gherkin mediated cell signaling uh, to the adjacent cells, that eventually leads to a signal coming from these follicle cells that ask nucleus to move to the anterior end of the oocyte. Now, here you see gherkin is also maternally contributed. You see bicoid, Oscar, etc. These are all the maternal genes being deposited. What I want you to go back and read in detail how gherkin and torpedo are responsible for movement of oocyte nucleus to anterior end of oocyte. So you should go and take it as an assignment, as a must read thing. Search for this content. This is within the textbook you have. So what, and then you will read all this. You see, we talked about Oscar stuff and Bitcoin and the maternal contributions, and you will see what these lines are, what this traffic is taking place, movement is taking place. You see these, what are these machines moving the nucleus as well as the maternally contributed mRNAs. So these are just the, because we just talked about Notch delta and JAK-STAT, but there are cell signaling events like hedgehog, wingless, and here they are written their role in the not only oocyte development, but also in the other process of organogenesis. So with that, we are done for today's lecture. Let me know if there are questions. Any questions? There are no questions. So let's take 10 minutes break and we will continue with our lecture about tools in development. Because of my sickness, my fever, we lost 2 of our lectures. And I have, Alhamdulillah, caught up the deficiency of those lectures without speeding too much. But we have to, after the break of 10 minutes, we will cover the tools. I want you to understand those tools because they are very important for you, not only understanding of developmental biology, but exam as well as the research papers which we are going to share with you. That was one of the reason I did not still send you the research papers, because I wanted to cover a few more tools so that uh, you easily understand the research papers. Okay, so let's meet in 10 minutes.